All right, before we move on to sample preparation sort of stuff, we're doing okay. We had some good conversation. Sounds like you guys are starting to loosen up, get to meet people around you, and or the coffee's working and donuts and all of that. Wonderful. Any questions before we begin sample prep?
There is a question. Does the depletion technique impact non-targeted proteomic study?
Non-targeted.
A depletion technique.
Depletion for like a plasma or serum, I guess.
I guess.
I mean, the depletion technique will make
Plasma proteins.
Yeah, for plasma proteins. Yeah, that eliminates albumin and some of those other guys.
Yeah, I mean, it affects it. And so there are certainly, there's certainly proteins that are going to bind to those abundant proteins and be depleted. Everybody's always, worried about that. I'm more worried that if we don't deplete, we're going to identify only the abundant proteins. And so you can always worry about something, but I don't worry about that. We have, some researchers that want to then reanalyze the depleted proteins. That's something we try to talk people out of. But I know, some people have done this. And so there are certain, cases where you're going to find an interesting protein that binds to albumin. But that's a really technically very difficult experiment.
Yeah. Yeah.
Are you limited to, a weird way to phrase it, biological samples? Say you wanted to take the mycoplasm from a cell culture experiment to analyze myokines or extracellular vesicles. Would that be possible with the size of those proteins?
Sure. Yeah, we do. I don't know, we probably have a couple of extracellular vesicle projects a month that come through, and it's difficult sometimes. The enrichment process, most of the kits don't work, don't work very well. Differential centrifugation is the best way to get enough material to analyze. But yeah, we do those all the time.
All right. Well, let's talk about sample prep here and just some, I guess some words of wisdom or from, I had to get rid of that thing up on the top. Maybe it'll go away here in a second. So sample prep goals. It seems simple enough to ask for a particular amount of sample. But how these are prepared determine experimental success. When the samples arrive on site, their fate has already been sealed by your hands in a lot of ways. And so, man, communication is hard. Communication's harder when you're doing it by Zoom and email. And the fact that we have biologists here, molecular biologists, we've got physiologists, we've got all, we got chemists, we have all gamuts of different types of things. And so the words that are coming out of my mouth and what are going in your ears and what your training has and what it means to you and all that kind of stuff, it makes it difficult. So, we just say, hey, we need these types of and so we have some basic assumptions that we make. If you're in a lab that does cell culture, we assume that how to do your craft and everything. But I'm going to go through some of these things that are sort of common things that come up from time to time. Number one, counting cells. We say 5 million cells or 10 million cells. And a lot of you guys are like, well, my plate is this big, and I know that when I grow it up to confluence, that's going to be about this many cells. And we sometimes will look at the size of the pellet that you send and we'll say, yeah, no, that's not, we only have 10% of that. And all that means is you say, well, you can't do proteomics on a million cells. We can. We just it's like a whoopee. It's like a comfortable blanket, right? We like to have more so that if something happens in the process, we have more to fall back on. So the proteomics usually is better if we get more sample from you. Also, washing cells. Almost I've done cell culture once or twice, so I'm on shaky ground here. But my understanding is that almost all cells are in some sort of fetal bovine serum media, and that is protein. And if that's all you want to see, then please don't wash that away and just send it to us, and we will see beautiful signal from that. So you got to wash all that away, and then use PBS to be able to that. And also, sometimes when you guys spin the cells down, if you don't do that gently enough, it'll cause cell lysis, and your cells will start to break, and then your pellet goes away. And these are all sort of techniques that you either know that or you don't. And then also freezing the cells. Being able to do that where you minimize the volume before freezing really helps. When you pulled off the PBS, maybe with a 100 microliter tip, and then you wait a minute and you come back with a 20 microliter tip, and you pull that last little bit down so and that helps. And then you drop that into dry ice or liquid nitrogen or something like that. Also, every once in a while, bioinformatics will go, hey, did anything weird happen with this in sample prep for this particular experiment? And of course, then sample prep goes, well, it wasn't us. And they look around, and then you go back to the investigator, and they go, well, actually, we grew up these cells on this day, and we grew up these cells on this day, or whatever. Those batch effects will then confound and make it really difficult to be able to pull out that biological variability that you're looking for. So, ideally, what you'd want to do is have all your cells grown up on the same day and kind of do all that at the same time. And there is sometimes, if you're doing a treatment, and I'd like some comments on this, but if you're doing sometimes we end up where the control turns out to have the greatest variability. And it's because you've waited a couple extra hours for your treatment, and your control has basically passed confluence. And so now it's not the same. One of these things is not like the other. And so all of those little factors that you can minimize in your lab will make your experiment a lot more successful at our end. Because again, the goal is to minimize that in-group variability. And to get a measure of it. So tissue samples, one of the biggest things for tissue samples is getting rid of the blood. Remove perfusion, which is, I think, actually a thing where you actually can hook up to an artery, and you can actually just push PBS through your organ of that, or just rinsing it, and then having a homogeneous sample. I mentioned before, kind of tongue in cheek about shoving half a rat brain in a conical tube and sending it to us, but it's happened. So if you're interested in rat brains, which I know there's at least one researcher in here who is, you'd want to send us specific parts of the rat brain, right, that you have pulled out, because the proteome is going to be much better because it's not going to be diluted down by having different cells from different parts of the brain. And we say we need 100 micrograms of protein to start. And that's because we have these standard workflows that make it very easy for us, if we're given that amount, to be able to use our standard workflows and go through our protocols here. But we also need to do a BCA, we do a protein quantification, and we like to use a BCA, which is a colorimetric reaction. And so if you send us exactly 100 micrograms, we're going to turn around and do a BCA, and sometimes we have to use 10 or 20% of your sample to do that. And then we have less. And then what if you were wrong in your estimation and you only had 50 micrograms instead of 100 micrograms? And this happens every single day, every single week in our lab. And so now we only have 35 micrograms of your sample. And so then we have to make changes as to what the protocol is. Now, can we do proteomics on 35 micrograms of protein? Absolutely. Will it be as good as 100 micrograms? I don't know if there's a big difference between those two, but we don't want 2,000 micrograms of protein, but we don't want 20 either. So somewhere around the 100 it would be great. Question, when should you send a lysate? When should you do the lysate and send it to us instead of sending us a cell pellet and that? It's not necessarily a rhetorical question. One of the answers that I have for that would be, if something that you're dealing with is going to be like, I don't know, something that's diseased, some tissue that's got, I don't know, hepatitis or something like that, we'd really like for you to make the lysate on your end and make that inviolable and just break it down into some proteins and stuff before you send it to us. What else? Rick, you had a comment about lysates. Is that good? Okay, that's good.
There is a question in the chat.
Yes, ma'am.
In cell culture, how to process extract the membrane protein and intracellular protein? Membranes and intracellular. Rick?
So that's not something we're going to do.
Right.
You know, I mean, there are kits for membrane preps that work reasonably well. The only thing that I would say is that if you're going to do that, there's variability, you know, in that, and so you need to increase your replicates accordingly because you know, anytime you're going to increase variability, you need to increase replicates. For extracellular matrix, I mean, that is almost a field of its own, and so I would say that expertise is going to have to come from the PI. And so we've certainly done that. You know, we've worked on projects where people are interested in extracellular extracellular matrix proteins. But that isolation is going to have to be done in your lab. That's not something we're going to do.
Yeah, that's a great point that I need to, yeah, we'll give you advice and all that kind of stuff. But if you're doing an enrichment or, you know, an isolation or something like that, that's all going to have to be, because then every once in a while we'll get somebody to say, yeah, I'd like you to do this and, you know, follow this paper or whatever, but everything takes that bad word is optimization, right? It takes optimization. So you do that in your lab, you get that fixed up. If you need to send us some test samples as you go along and do that, we're cool about that. You know, run a couple samples here, you know, see how it's going. And then, you know, get your experiment done there. Yes, sir.
How long can we store the lysates from we're ready to be ready for your process here? For example, how many days?
At minus
Weeks,
eight, at
months?
minus 80, I would say,
Yeah, I'm a chemist, not a biologist. So to me, if it goes to minus 80, it's good forever. You know, I store peptides out on the lab bench. And so, no, it's, you know, I think a year is probably fine. You know, we've certainly identified, you know, had a lot of projects with stored samples. And so I wouldn't worry about that. I think that's one of those things biologists worry about too much. You know, I worry about the seconds before it goes in the freezer a lot more than I worry about how long it's actually in the freezer.
Can I ask a second question? For example, we handle the hearts, and for that sample, once we collect it before we lysate, is a suggestion to not store at minus 80, but in the liquid nitrogen. So which one you prefer? Liquid nitrogen versus minus 80 for tissue?
I think it doesn't matter to me. You know, I would just hope that it gets perfused before it does that. So that's more important than anything, is trying to just get rid of as much blood as you can.
Yeah.
But yeah, I guess we probably haven't ever done a study where we've looked at that. We've done various studies on serum, on how they're stored, and you know, a lot of variability of serum is how long it sits in a vial before it gets processed. But I don't think we've ever looked at tissue and how it's stored. It's a good question.
Yeah.
Patient samples, it is very difficult to perfuse. I mean, we cannot perfuse them, and they are usually very bloody.
Yeah, so with that, you know, I'd recommend a protocol where the tissue section at least gets rinsed in PBS before it is snap frozen. And so that, you know, and that I realize it's, you know, requiring training of probably somebody in a surgical suite that are, you know, doctors are the worst ones to train. But that's
Liquid,
probably the
hydrogen peroxide. They just pour. That's what I've been told.
I don't know, but I think, you know, any rinsing is going to be better, right? So I mean, that's one of the biggest problems that we have with human samples is the amount of blood. And you know, with tumor, it's difficult because, you know, these are all very well vascularized tissues, and so there's a lot of blood there, but it's going to impact the variability.
Thank you.
Okay.
Sure. Yeah. I just use that ubiquitously as something that doesn't contain fetal bovine serum. Yeah.
Right. Good questions. It's good to get all that stuff out and on the front end here. So I wanted to show you, I wish, I don't know how to make that thing on the top go away. Stephanie, do you?
You just wiggle the mouse around below it, click somewhere. Nope. Okay, it's just there with you forever.
Do I need to wiggle while okay, all right. This one? Hide floating media control. Oh, thank you. You guys rock. Okay.
All right,
So-
you get a free donut for that one. That's good.
There's no, it doesn't. Yeah, okay. So I just wanted to talk about our BCA just a little bit and what it looks like. Some people were even asking me out in the hall, you know, about a nano drop or different ways of measuring protein and that. And we like to do the BCA and the reason is because it is a colorimetric reaction that is a correlation between the color purple. Up there on the top, you can see the standards there. If you've ever done this before in a plate reader, it takes time to do it. It takes about an hour to make up the reagent, you know, and incubate it for 30 minutes and then collect your data and all of that. But this is the best way that we have found for us to be able to get our, it's not perfect by any means, but it's the best way for us to be able to get the loading, to get all the samples to the same concentration and to have a pretty good idea of what the protein amount is so that when we add all of our reagents to do our alkylation and reduction and digestion, all that kind of stuff, that's pretty good. So I'm not trying to hurt anybody's feelings, but what we want you to do is we want you to measure your protein quant on your end before you send the samples. That you need to do that because you need to know how much you're sending us. Well-
Dennis, isn't nano drop easier? Why don't we do that?
Well, because there's interferences, other things that are in your sample that will absorb at that one wavelength that it's measuring. And so the reason why this is better is because that purple color, I mean, there is a long, a pretty well detailed list of things that can interfere with this colorimetric reaction. But for the most part, it's the peptide backbone that is then causing that color. And so we have a much better shot of getting an accurate protein amount. But we're going to turn around and we're going to measure the protein quant of everything that you send us anyway. Because there's just way too much work that we're going to do. And we don't want to get all the way to Stephanie and she does the bioinformatics and goes, what in the world happened with this? And we go, well, the investigator from University X said this was the protein quant and we just rolled with it. Because you're not going to want to hear that when we give you your data and it looks like it bad. So anyway, that's what we do. Let's talk about lysates just a little bit because this happens from time to time. This picture over here, we've had people send us samples before that said, we've already made the lysates and from that lysate, we did our own protein quant and it looks literally like a peach milkshake there, right? So just so you know, we would have to take that, and Robert is going to probably tear me up on this, but I would say this is what a lysate more closely, where we finally, after filtering and spinning and all of the things, we use a probe sonicator and everything, getting it down to where it is, it's not colorless. Usually there's a little bit of a color to it. Sometimes they're clear and colorless, but we can't have anything that's got chunks or bits of stuff floating around there to do a protein quant on, right? Because you're not really getting the real protein quant. It's not a homogeneous mixture. It's a heterogeneous milkshake. And so you're not going to get that. So that's something that we spend a lot of time on, and I mean, it's one of those steps in the process that, boy, we'd love for all of you guys to just send us the lysates, and you do the protein quants for us, and we could believe those, and all we have to do is just go start our standard workflow. It'd be a lot easier, but we probably spend, I don't know how much time in man hours per sample, but a lot, because that is the secret of doing good proteomics, is the sample prep. Not to downplay the mass spectrometry or the bioinformatics, but
You're
if
just making my job easier.
that's exactly right. That's exactly right. So having all that stuff done on the front end. So we were talking about this, I think you were talking about biological fluids. So here's the, and it's hard to say, but this is the upside down plate that contains a bunch of plasma samples. And you can just see here a zoomed in shot of just the rainbow of colors that we have here, because it's hard. It's hard to draw these things and not have hemolysis in there and have blood in there. And so that doesn't mean that it's ruined. I'm just saying it's just another challenge. It's another factor that we have to, that this sample is going to have. The mass spectrometer is going to be occupied looking more at blood proteins and peptides than your sample. Do I have anything else I want to say there? So that's why a lot of times when we do that for biological fluids, we like to deplete the top 14 most abundant protein. I think that's the most. I don't know if they've made it anymore. It used to be 2, and then it went to 12 or 10, and now we're to 14 most abundant proteins here. So sample prep goals. So here's a violin plot, again, just like a zoomed in shot. And so it's like a top down view of the data. And so what you're looking at is, it's like you have down here, this is the normalized intensity. So this is the really low intense things. And then these are the really high intense things. And so you've got the median right here in the middle, and you see where that little line is. And so if after the data gets normalized, all these little lines, the loading should all be about the same. And we're looking for equal loading of the protein on the front end, so that on the back end, this normalization, will do it just right. But the basic assumption that we have is that in global proteomics, the overall abundance of the proteins in tissue or a cell culture or whatever will not change during the experiment. Even though you're doing a treatment, even though there's a knockout, even though there's a disease or something, the cell still has to function. There's still a whole bunch of stuff that has to go on. And so as long as it's a living, viable cell, there's just going to be a small percentage of proteins that are going to change, and most of them are going to stay the same. And so the overall abundance is the same. That is one of our overall assumptions here. So sometimes you get, there it is. So this is just another example where somebody sent us some data, and one of their samples seems to be abnormally different from the others. It's like, what was that, Sesame Street, where you say, one of these things is not like the other. You remember playing that? Maybe you guys grew up in a different generation. And so that just means that it's going to be harder for us to be able to compare the biological variability between the blue samples and the red samples and everything if there's so much variability within the red samples themselves.
Yeah, that would probably be thrown out. And this is also why we want replicates.
Thank you. That's right. And if you'd only send us 3, there's lots of weeping and gnashing of teeth. Yeah, you have a question?
Yeah, so just to your point there, Stephanie, I was wondering this, like if you do a Western blot, you're only looking at one protein and you get all your points are nice and clumped here, except for this one out here. And then you look at it and say, oh, that's probably a statistical outlier. Can you do that, if you're looking at 4,000 proteins and you get this one animal that's just, sticking out there?
Yeah, that happens. That's why we want to have at least 5 replicates or something like that in the experiment in case there is one like this, because a lot of times something like this is going to be plasma serum samples where you got a lot of hemoglobin or a lot of high abundant proteins, something didn't get depleted. That might be part of this one. It could be any number of other technical issues, but I want to be able to have the ability to remove a sample that is different and still do the statistics on it.
Okay, I'm going to move on. So this is a chart over here that's showing you a particular protein. And each one of those dots represents its measured intensity in a different sample. And then we're comparing these different samples together here. And again, the goal is we want to limit, this is the variability, the in-group variability that we want to limit. So for the blue versus the red, you can see that we have a certain amount of variability there. And what we're trying to do is minimize that or get a measure of it, right? I mean, sometimes you've got some sort of isolation or something that you're doing that's going to cause a certain amount of variability, and that's just, it is what it is, right? But you want to try to minimize that so that it's as small as possible. You can see the variability, the in-group variability is a lot smaller over here. And then we're looking at the biological variability between the groups there. We're trying to maximize those guys by how we do our experiments. Okay, so just to give you an idea, because we talk a lot about, this is the what a chromatogram looks like of a normal lysate. Okay, it's you might remember chromatography maybe back from your undergrad days where you would have baseline resolution and you'd get one nice peak that would come out, and then a minute later another peak would come out. I mean, it's just in a 45, 50-minute gradient here, we have just, that stuff is just flying out. It's just amazing how fast the data is coming out and being processed here all at one time. And so we're trying to there's a certain dynamic range or a certain range of stuff that we can get good data on. And so we're trying to, people are always asking, well, how much can you actually load on the instrument? Well, if you're talking about a lysate, we can load about a microgram. But if you had a single protein, we wouldn't want to load a single protein, the peptides from a single protein, because each peptide would have this huge amount and it would go way off scale, right? So here's what it looks like if you had hemoglobin contained, or this is a bloody sample. And so, I don't know, maybe your trained eye, you can see the kind of the difference here. All of these peaks that are rising up out of there are the peptides from hemoglobin. And so you can see that the level where all the other proteins are much lower there. And so there's going to be some things that we're going to lose because we're so distracted by all of these other guys up there. And I'm picking on the hemoglobin today. It's usually it's albumin. Okay, I'm saying albumin is a culprit, and hemoglobin as well. It just happened to be what was in the lab last week when I made these slides up. Okay, we're almost done here, and then we're going to kick it over to Stephanie here. So sample variability. We want to make sure all the samples are treated the same. Now, Robert's got much better eyes than I do. And so when he looks at these up here, he looks over the one on the far left and he goes, that one's kind of got a yellowish tint to it compared to the other ones, right? But then one of the samples look like this, and one of them look like that. And this is all in the same sample group, right? So I'm not trying to pick on anybody, and I know you guys have experimental challenges in your lab. But the variability, just if you can see it with your own eyes before you even start the sample prep, you know that we're going to have some in-group variability here from that. So the more that you can minimize that and press upon your staff and the people that you work with about that.
And also take a note of that, because when we see the analysis and we see like a PCA plot and that one's looking a little bit different, but we have a reason for why it's separated out. So it helps with interpretation.
Yeah.
It may not necessarily get thrown out of the analysis because you want that variability kind of within your experiment. So there's different things that we're going to look at the QC.
And all of those samples all those pictures were taken to put into the file so that, 4 to 6 weeks later, when we're doing the bioinformatics, we can go, there's a lot of variability here. And then we look back and there's a note there from sample prep that said, yeah, it looks like they had some issues here with some variability of the samples. So let's just, as a summary here, we want to limit the variability in your lab, when you do your experiments there, and be able to take a close look at that. You want to be able to measure the amount of protein that you send us, so that that's really hard, but there are times where you don't send us enough protein. One of the, and we have to ask for more sample because of that. And then also just the communication and asking questions. And here's probably the most important slide of the whole thing is that if that's a 1.5 milliliter centrifuge tube, that is about the amount of sample, tissue, cell pellet that's all we need is that much. Okay, just that little bit right down there. That's the sweet spot. Everything goes, everybody's happy with that. Yes, sir.
I don't know where it counts, but.
Yeah, about 50. Yeah, sure. What would you say? I'm pointing at Robert here because he looks at this every day. About 50. Yeah. Okay.